with the class. So this is another one-shot revision session, and in this lecture we are going to revise unit number 6, that is your plant physiology. We have already covered unit number 1, 2, 3, 4, 5, 10, 11. What else? Unit number 9 uploaded. Yeah. Now we are doing unit number 6, that is your plant physiology, right? So plant physiology, very scoring, very interesting and very predictable unit as well. By predictable, I mean sometimes, like almost every year, there are few set topics from where you will get a question. For example, plant hormones. This is something from where every year in one format or the other, there is one question, right? Then you have nitrogen metabolism also being repeated a number of times now. Followed by that, you have photosynthesis, you have respiration. All these topics are something from where every year in one format or the other we get the question. And it's a very interesting unit as well, because see, when you're able to correlate things, it becomes easier for you to revise. It becomes easier for you to remember the things. And plants are something that are very much there in your surroundings, so it's comparatively more visual, I would say, and visual makes more better, like a much better memory, and you're able to correlate more, right? So make sure that whether you're from botany background or you're from zoology background, try not skipping plant physiology. Even if you're from zoology, try doing some important topics, like at least few topics of plant physiology, which are very, very favorite. Okay. So favorite of CSIR, GATE, and all the other examination. And you can utilize these like one-shot revisions, not just for your CSIR, but for your other exams as well, right? Let's begin from the basics of plant physiology. So when I talk about the basics, first segment is about the water potential. So water potential is what? It is the potential of the water to perform a specific functionality. There are 4 different components of water potential. What are the 4 different components? You have solute potential, you have pressure potential, you have gravitational potential, and you have matrix potential, right? In this, the gravitational potential and your matrix potential, these are taken as almost negligible or zero. So as a result, water potential's final formula turns out to be the solute potential and your pressure potential summation, because these two are almost negligible. Then we have water potential being, or specifically, your solute potential is always negative and your pressure potential is always positive. It can be positive or negative, both. Then we have something called as incipient plasmolysis. So incipient plasmolysis means when the potential of the cell is equal to the potential of the solution in which it's kept. Imbibition has a very low water potential. What do you mean by imbibition? Try defining imbibition, everyone. Then aquaporins regulate the water uptake, and over here there's a protein called as PYQ that gets active. Then you have apoplastic flow, that is blocked at the Casparian strips. So when you look into the cross section of your root, in that there are various different compartments. One of the compartment or one of the strip in the name of Casparian strip, and there are 2 different types of flow that can be there. One is called as the apoplastic flow and the other one is symplastic flow, right? Apoplastic flow is what? Apoplastic flow is that when the transportation is occurring via the cell membrane or the upper layer, and symplastic is that when it is occurring via your cytoplasmic connection, right? This cytoplasmic connection is given the name of your plasmodesmata. We learned about this in the plant in your unit number 4, that is cell signaling as well, right? Then you have, see over here, the water potential and the transport mechanisms. So we have the transportation mechanism, first one as diffusion, then you have the second one as osmosis, then we have the bulk flow, then we have the active transport and the secondary active transport. Diffusion is what? It is random movement of the molecules down the concentration gradient, that is from an area of higher concentration towards an area of lower concentration, right? Then you have osmosis, that is the movement of water across a selectively permeable membrane. So when the movement is occurring but via a selectively permeable membrane. Then we have the bulk flow. So bulk flow is in which you have the driving force being the pressure gradient. Then we have active transport and secondary active transport. So active transport is the movement of the solute against the concentration gradient. That is when the movement is occurring from an area of higher concentration towards an area of lower concentration. And in this process, there is requirement of ATP. Then you have secondary active transport. So it is the movement of the solute that is coupled with the movement of an ion down its gradient. So when I have my movement occurring of a molecule along with an ion, right? So when the solute's movement is coupled with an ion together, it is called as the secondary active transport. Over here you have the electrochemical gradient being the driving force. Over here the energy requirement is indirect. In case of active transport, it is required, like primary requirement is the ATP only. Bulk flow, no ATP required, and in osmosis and diffusion as well, because all of these are occurring down the concentration gradient. So it is occurring passively without any ATP requirement. Then we have the root pressure, or something called as guttation, right? So because of the root pressure, you get to see the guttation being there. What is guttation? So if you see a leaf, and on this leaf, in the early morning time specifically, you get to see the edges of the leaf having water droplets on it. So these water droplets are because of the root pressure that gets developed during the entire daytime, during the night time, right? Then the increase in the transpiration is with temperature, light, wind, vapor pressure development, all these things. Then you have cohesion. Cohesion tension theory is the one that helps to maintain the water column. Then what is cavitation? So it is embolism. It is repaired via your root pressure. Then Arnon and Stout, they gave the criteria for any element to be considered as essential, right? Write down what are the various criteria for which we cause, or for which we make, or for because of which we consider any element to be essential in nature. Try defining it. Then carbon, hydrogen and oxygen makes up around 90% of the entire dry matter. Then you have the water potential. So this is solute and pressure potential, because other two are almost negligible. Then driving forces, the gradient for water potential. And what is the transport pathway? Cells, tissues, soil, water, atmosphere, continuum. So there was a question asked based upon this thing as well, that what do you mean by the soil, plant and atmosphere continuum? So this is something that is defining your transpiration or the water loss, right? Because the soil absorb via the soil, the plant absorbs the water, and from the plant the water gets evaporated out, right? Then you have the water uptake and the movement in the root. So it is via the xylem. It is a gradient of your water potential, which is negative in case of your xylem. And over here you have apoplastic and symplastic pathway. Then you have the xylem transport. So the bulk flow of the water and the minerals being there. In this we have the negative pressure, that is the tension from the transpiration at the leaf surface. And over here you have the cohesion tension theory, right? Because, or for the water to be transported from the root towards the leaf area, like against the concentration gradient, there's a capillary action that gets utilized. So this capillary action, with the help of this capillary action, there's a cohesion tension theory which explains this against the gradient or against the gravity movement of the water. Then you have translocation. So movement of your photo assimilates, that is the photosynthetic contents that is getting produced. So movement of the photoassimilates is explained via the phloem translocation, then which is, and the driving force over here is the positive pressure, that is the turgor pressure from the phloem loading pressure flow hypothesis, and the transport is from the source area towards the sink area, right? Then nitrogen deficiency can cause chlorosis. Very important slide. Okay, very, very important slide for you. Nitrogen deficiency can cause chlorosis. Potassium deficiency causes purpling. Then potassium regulates your, sorry, phosphorus deficiency causes purpling. Potassium regulates your stomatal opening. Iron is very much essential for your cytochromes. Manganese deficiency causes the brown spots on the leaf, right? Then you have molybdenum is essential for your nitrate reductase, and zinc is required for your oxygen biosynthesis. All these are potential part B questions, right? All of these are potential part B questions. Then LHC, light harvesting complex 2, contains chlorophyll A, chlorophyll B plus carotenoids. Now these are the pointers for your photosynthesis. Then TS2, that is photosystem 2 is P680 and photosystem 1 is P700. Hill reaction is the photolysis of water. Non-cyclic photophosphorylation, that is your Z scheme that we talk about. In this, ATP and NADPH both are produced, and when we have only the cyclic photophosphorylation being there, only ATP is produced in. Rubisco has oxygenase and carboxygenase, both the activities. Depending upon the concentration of your oxygen and carbon dioxide, you get to see the functionality being there, right? C3 has high CO2 compensation point and C4 has lower one, right? Then photorespiration is in which 3 different organelles are utilized in the entire process, in which there occurs unfixing of carbon dioxide. So first is chloroplast, then peroxisome, then mitochondria. You can remember this mnemonics as well. And in the C4 plant, it uses PEP carboxylase as the enzyme. CAM plants, in this the stomatas are open at the night time. In the Z scheme, the water is, like, there occurs photolysis of water, and as a result of photolysis of water, we are having the production of NADPH in the final segment plus water plus ATP being there. Then transition state balance is there between the PS1 and PS2, which maintains the entire functionality in the Z scheme or in the entire photosynthesis light reaction, right? Then you have photoinhibition that damages the D1 protein. Write down whether the D1 protein is a part of PS1 or PS2. In photosystem 1 or in photosystem 2, in which of the photosystem do you get to see the D1 or the D2 protein, D1 protein as a part of the segment? Then Kelvin cycle activated by your thioredoxin. RUBP, in this regeneration is requiring ATP. The shade plants saturate quickly in the light curve, and P680 is the strongest biological oxidant. This can also be a potential question. Quantum requirement is around 8 to 10 photon per oxygen molecule release. Then Kranz anatomy is there in C4 plants. What is Kranz anatomy? So there are 2 different things. First thing is the arrangement of your bundle sheet cells and the mesophyll cells in circular width like structure, and second segment is presence of granule and agranule chloroplast, right? Write down in detail which particular cell have granule chloroplast and which one has agranule chloroplast, and what is the reason for the same, what functionality or what function does get imparted because of this. Then chlororespiration is via NDA2 PTOX. Glycolysis is there occurring. Glycolysis occurs in the cytosol. Then when we look into my ETC, electron transport chain, so in the electron transport chain, there is pumping of H plus ions happening in. Complex 1 pumps the 4H plus. complex 3 pumps 4 and complex 4 pumps 2 of them. So when you revise, you'll get to, like, you'll be able to recall it better. Cyanide inhibits your complex 4. So there are various inhibitors for various different type of complexes. So make sure that you remember all the inhibitors and try writing down any inhibitor that you can recall, any inhibition that you can think about it right now in the comment sections. Then AOX bypasses the insensitive to cyanide. Then we have the main photosynthesis and the photorespiration, mnemonics being there. I've already like shared a lot of mnemonics with you all. These are also going to help you. So the photorespiration cycle, which is the C2 cycle, in this occurs in chloroplast, peroxisome, and glyoxysome. So over here the mnemonics is car parking garage, so CPG, right? Then you have, or majorly it is your C2 cycle is CPM, glyoxysome, okay, which is specifically a part of your mitochondria only. Correct. So then you have the chloroplast, peroxisome, glyoxysome. So in the chloroplast you have RUBP, oxygenation happening in, peroxisome, glyoxylate oxygen formation happening in, and in your mitochondria you have the serine formation being there, right? Then in the C3 pathway there are 3 stages of the Kelvin cycle. First is oxygenation, then you have reduction, and then you have the regeneration being there. So remember, entire C3 cycle along with its energetics. Energetics means how many ATPs are being produced in which step, how does the ATP get produced, what are the other byproducts, what is the net output. All these things are important. What enzyme is participating in, what is happening in the process. So for example, in case of your carboxylation, you have CO2 fixation to the RUBP. Then you have key enzyme. So Rubisco is the key enzyme, that is ribulose 1,5-bisphosphate, carboxylase and oxygenase both. Function is for carbon dioxide getting combined with RUBP to form the 3-PGA. And over here you have what is being consumed and what is being formed. Same goes for reduction and regeneration as well. Make sure you remember it. Then we have the comparative analysis between C3 plant, C4 plant, and the CAM plants being there, right? 3 different segments are here: C3, C4, and the CAM plant. So in the case of my C3 plant, the first CO2 product is, the first carbon product is the 3-PGA. Over here it is oxaloacetate. Over here it is again oxaloacetate. Then primary oxygen fixing enzyme is Rubisco. Over here it is pepcase and Rubisco both. And over here it is pepcase in the night time and Rubisco being there in the daytime. Then anatomy, standard leaf anatomy. Over here you have Cran's anatomy. Over here you have standard leaf anatomy with larger vacuoles being there. Then temporal separation is there only in case of CAM plants, because CAM plants have something called as scotoactive stomatas being there. That means those stomatas that are open during the night time, which causes your separation of or concentration of CO2 only during the timing when Rubisco isn't available, which leads to the decrease in the C2 plant being there, or which leads to decrease in the C2 pathway getting activated as a result of the oxygen concentration being higher and oxygenase format or oxygenase segment of RUBP getting activated. Then spatial separation, there's no spatial separation. Over here there is, and over here there is no. Photorespiration levels, optimum conditions, and the examples are also given to you. It's a very important slide. You can easily get statement wise question. In the recent year as well, we got a question from the CAM pathway. So remember, all the pathways are important. Then you have C4 cycle. What are the molecules, how many carbon atoms, and what is the location? All these are important. Over here you have the pictorial representation of the same. You can go through it. Then you have CAM pathway. So this is crassulacean acid metabolism. They use temporal separation to minimize the water loss. Then diagram, try to like over here, no, it's not here. You can just make sure that you revise it and keep a note of that how does the temporal separation looks like. So nighttime, stomata is open, process or location is the mesophyll cell, then you have the key reaction happening in, then you have during the night daytime the stomatas are closed, so your mesophyll cells location same, and over here the CO2 is being released, malate gets broken down, and as a result of the malate being broken down, CO2 is released, and then the C3 pathway can be activated with your RUBP having carboxylase segment activation. So remember, Rubisco has both carboxylase and oxygenase activity. Okay, so because of having both of these activities, and the which particular activity will get activated depends upon the concentration, because the active site for CO2 and O2 is similar. So if the concentration of oxygen is higher, oxygenase will get activated. As a result, your C2 pathway will get activated. If concentration of CO2 is higher, in that scenario, you will be having your oxygen carboxylase activity being the higher one. So as a result, you have your C3 pathway getting activated. So this temporal separation helps the plant to avoid C2 pathway and to also conserve more water.